Good morning, everyone, from my behalf as well. My name is Hanna Vuorenpää. I'm a postdoctoral researcher in Tampere University. You might have noticed us from the violet t-shirts here today or in this event, and I want to thank the organizers for giving the opportunity to present our results here. So I'm going to tell you today about the vascularization and cellular rearrangement in bioactivated gellan hydrogels. First, something about our consortium. So I work in KOIBOK. It's a center of excellence in body and chip research, and we are composed of 6 different research groups. So we have 3 groups working with human stem cells. Then we have a group for biomaterials, a group working with sensors, microfluidics, and then imaging and numerical modeling group. So you can see here our website, so please go ahead and take a look. There's nice videos, animations like this. You can check our work in there. So what do? We develop body on chip platforms to model systemic effects of ischemia. So you can see here animations of our target tissues. So we have chosen a couple of those, and we are focusing on cardiac and brain ischemia, as well as hypoxic adipose tissue, and of course moving on to study ischemic responses to all of these organs. We also want to extrapolate from in vitro to organ level and to comorbidities via computational modeling. So you can see here animation from the target tissues, and we have chosen the approach to include hydrogel within our cells to provide the 3D structure and the appropriate environment for our cells. So this is what we do currently, sort of in a big picture. So if you first take a look at this left-hand side, you can see studies ongoing with the vascularization. So we have already published this perfusible vascularization last year, and now we are moving on to vascularized adipose tissue, bone tissue, as well as skeletal muscle tissue that we heard in the previous presentation. In the middle part, you can see neurovascular interaction, but this is something that we are certainly aiming for. So to combining the vascularization and the innervation within our target tissues, and this came out, I think it was actually last week, so please go ahead and take a look at that if you're interested. And on the right-hand side, you can see ongoing work related to innervation. So we have work with innervated cardiomyocytes. Now a lot of work, if you can see, with the innervation of adipose tissue, and big focus currently is on the peripheral neurons. So like I mentioned, we want to combine vascularization and innervation within our target tissues. This is our organization. So you can see in the upper row our PIs, so our professors. Like I mentioned, there are 6 research groups with the biomaterials and tissue engineering. There's hard group, micro and nanosystems group, adult stem cell group, neuro group, and computational biophysics and imaging group. And we are organized so that we have, beside the PIs, we have the vice PIs that support the work of the PIs and sort of act in between the PIs, the professors, and the students and the researchers. And we are also very active with the vice PIs, and actually we just got acceptance to our review that is about building blocks of heart on a chip. So please also check that. You can find it, for example, with my name and building blocks of heart on a chip. So these are the research groups that work with this study that I'm going to present you here today. And here's the focus of this presentation. So I'm talking about bioactivated gellan hydrogels that affect cellular rearrangement and cell response in vascular co-culture and also in vivo. So we are talking about modified gellan hydrogels. There is 4 of those, 4 different hydrogels. I'm going to explain the details in the following slides. And we did 3 different studies with the mechanical analysis, in vitro vascularization study, and also an in vivo subcutaneous implantation study. And I'm going to tell you about this. I hope you can see this in vitro vascularization part. So this is our experimental setup. Like I mentioned, we have been using gellan gum. And why gellan gum? Because well, it is bacterial derived, it's animal free, which usually means smaller batch-to-batch variation between the hydrogel batches, and it's also already approved by the regulatory authorities, so EMEA and FDA for drug as well as food applications. So it's really safe material to use and quite easy to handle as well. So if we take a look at these different modifications that we have, so in the figure A we have sodium purified gellan gum, quite simple modification. Then moving on to B, with purified gellan gum that is functionalized with charge neutralized chimeric avidin and further combined with bioattenuated fibronectin. Then here on C, we have oxidized gellan gum that is coupled to carbohydrate modified gelatin, and according to this CDH modification we call this gela-CDH. And on D, we have oxidized gellan gum that is coupled to adipic acid hydroside modified gelatin, and again we have an acronym for this according to the modification with gela-ADH. The figure E is also important as we show the properties of our hydrogels, so they are self-supported and they are transparent, which is super important when we want to use these hydrogels in cell applications, in MPS, and we should be able to image these structures. So in this study that I'm going to present here today, we performed a co-culture of TFB tagged HUVECs, so human umbilical vein endothelial cells that were co-cultured with ASCs, so adipose tissue derived stem or stromal cells to induce vascular network formation for 2 weeks. And this is what we saw. So we saw quite a big difference in the capacity of these different hydrogels in supporting the vascularization and cellular rearrangement. We can first say that this co-culture of these endothelial cells and stromal cells could be maintained in all of these hydrogels for the 2-week culture period. But you can see big differences, like I said. So if we first take a look at these purified gellan gums, either with or without the avidin, we can see that the cells are rounded in structure. So only sporadically you can see a small elongations of these, actually both cell types. So this hydrogel is not really supporting the cells for the rearrangements. But moving on to gelatin modified hydrogels, you can already see there's a lot of more things going on happening. So we can see clearly arrangement of these cells, elongations, and so in here in red you can see alpha smooth muscle actin positive cellular network, and on green these are HUVECs that are forming quite small vascular structures, but you can still see some. So in my opinion as a cellular biologist, this was the most significant finding of this hydrogel study, that we actually saw formation of this alpha smooth muscle actin positive network instead of the vascular network that we were sort of expecting and hypothesizing. And out of these 4, we can see that this gela-CDH is giving the best support for the cellular rearrangements. We did a bit of statistics, not very extensively, but we wanted to have some in the study. So we measured the alpha smooth muscle actin positive cell network formation in these different hydrogels. So these are signals from these alpha smooth muscle actin positive cells. And you can see clearly differences between the different hydrogels. if we first take a look at these purified gellan gums, you can see quite a modest formation or cell coverage of these alpha smooth muscle actin cells, whereas again the gel CDH is performing, giving the best support for the cells to rearrange. Also gel ADH quite good, but still a significant difference between the gel CDH. Then what about the vascular network formation in these different hydrogels? Here we measured the endothelial cell area, similarly to what you saw in the previous slide. So if we first again start from the purified gellan gum, we can see actually quite a fast loss of endothelial cells with actually no recovery. A bit similar trend with the avidin modified or purified gellan gum. However, it's more stable when the culture continues. The gel CDH, we can see a small decrease in the EC coverage at first, but then we can see an increase from day 6 onwards. Gel ADH shows initial climb in the EC area, but remains steady from day 4 onwards. And let's see if we can make these videos work. I had them in a sort of wrong format, so it's okay. I can show. That one, please. So, thanks. So, we wanted to study or sort of search for optimal imaging methods for our hydrogels and to study the cellular rearrangement. So this is OBT, imaging from our vascular networks or the whole cellular network, and you can see that we can get quite a nice sort of mesoscale imaging from the OPT, so the optical projection tomography, with our hydrogels. So here is again the gel CDH chosen as the best one for this imaging, and we were very happy to see that this method works, and we can see the sort of 360-degree arrangement of our cells. Thanks. And here's another one. So here the endothelial cells are stained against the von Willebrand factor, so showing the small vascular structures that have been formed in the hydrogel. However, they are quite modest, but again, with this method, we can see the whole picture of the large scale cellular rearrangements in our hydrogel. And to conclude, we investigated 4 different hydrogels with cell attachment sites provided via gelatin, and that gave us modest vascular network formation or support for the vascular network formation. The gelatin-containing hydrogels strongly supported formation of alpha smooth muscle actin positive cellular network, and like I mentioned, in my opinion, this was the most significant finding of this study. And out of these 4, we could see that the gel CDH was performing the best, quite clearly, at least from the cellular perspective, and we were happy to continue with that with the imaging method. So the optical projection tomography could enable mesoscale or large-scale 360-degree imaging of our cellular networks in these gellan gum-based hydrogels. So we can conclude that gellan gum-based hydrogels can be modified for tissue engineering applications. However, further investigation is needed on the biomaterial cell interactions and following responses to provide optimal material for target OOCs. I want to acknowledge all three groups that were part of this study. So our adult stem cell group, computational biophysics and imaging group, and biomaterials group. And special thanks goes to Christine Gehring, who actually last week successfully defended her PhD thesis. I don't know if you can see this. This is Christine's book, and she also printed us this nice hydrogel figure that we could take a look at during the dissertation. And we are funded by Academy of Finland, and if you want to have any information on our consortium, please contact me and Vuorinen, who is there. Go ahead and contact her, or in the current study you can contact me. Thank you for your attention. I'll be happy to answer questions.
Thanks, Hanna, for a very interesting talk. Questions? Okay, then I would have one to start with. So you said you were rather surprised that you didn't see the HUEX grow and you saw the alpha smooth muscle actin being expressed.
Yes.
I think you didn't expect this, so what do you think is the cause? You said you're speculating at the moment?
Yeah, we are speculating at the moment, but at least I think one characteristic of the hydrogel, so the stiffness likely has an effect on how the cells can rearrange themselves. So maybe we have a too stiff material for the HUVEX, or the endothelial cells, to arrange, and then sort of the smooth, alpha smooth muscle actin positive cells is a good environment for these. So this is one speculation that we have, but we don't have the details on what is going on there actually.
And do you plan to seed muscle cells just to see if you're able to make them differentiate on the hydrogel then?
It's a good idea. It's actually a very good idea. We don't have those plans yet, but let's see how we continue. Thanks for the suggestion.
Any other questions?
Hello, Hanna. Thank you very much for your talk. I was wondering, now that you were mentioning about the stiffness of the gels, have you characterized the gels in terms of, well, mechanical properties and cross-linking degree?
There is some characterization of the mechanical properties, and this is done by Christine Gehring. I don't know the details of those characterizations, but you can find them in the publication.
Okay, if there aren't any further questions, I would like to thank Hanna for the great talk, and now